Now children, the next topic that we have to study is the chemiosmotic hypothesis, and by this we are going to understand how ATP is synthesized, that is how ATP synthesis take place or occur during cyclic and non-cyclic photophosphorylation. So we have to study as simple as it is shown in previous, that is in cyclic and non-cyclic. It is not so simple. It is a complex process, and how it is occurring, that we have to study here. So for this we have to, I am making a single thylakoid. This is a thylakoid that I have already told you, that is the double membrane structure. The thylakoid is a double membrane structure. So this is a structure of one single thylakoid. So this is the outer membrane, this is the inner membrane, and the space which is left, that is the lumen. And we all know that the thylakoid is present, and outside the thylakoid the stroma is present. Okay, a jelly-like matrix is present all around the thylakoid. I am just making a single layer. So this is what? This is stroma. Okay, around the thylakoid a stroma is present. Now when we have talked about the cyclic, non-cyclic photophosphorylation, there I have told you that a site of photolysis of water lie in the inner membrane. Suppose this is the complex, this is the inner membrane where the complex of photolysis of water is present. So this is the complex for photolysis of water. Okay, one thing I am already told you there, and that is that the enzyme, that is NADP reductase is present on the outer membrane of thylakoid. Means ki the NADP reductase will lie somewhere here, and it is NADP plus reductase. It is a enzyme that will going to reduce the NADP into the NADPH2 that is formed during the process of cyclic photophosphorylation. Now these two things is clear to you, that is it is present in inner membrane and it is present in outer membrane. Now we talk about the two photosystem. Okay, so one photosystem will lie somewhere here. This is photosystem 2. Okay. And next photosystem will lie here, and this is photosystem 1. These two photosystems consist of that antenna complex along with that primary acceptor. All will lie here only. Okay. This much is clear. Now, children, what happened when the light falls here? Okay. When the light is coming here, it was taken up by the photosystem 2, that is having absorbed light of 680 nanometer. Now, remember the non-cyclic process. Then what happened? In that, it was the electron get excited and get accepted by primary acceptor, which is present in photosystem 2. Then after that, the electron loses its energy and it will enter into the electron transport chain, and there it was first accepted by plastoquinone, then it was accepted by cytochrome complex, and then it passes to plastocyanin. So I am not making the whole process, but I am making the thing that is it is accepted by cytochrome, and then it is passed to PC, and from plastocyanin it is passed to this photosystem 1. Okay, this will happen. The cytochrome, actually I have mentioned there that it is an electron acceptor, but actually it is not an electron acceptor, it is an electron transporter, because it is accepting electron from plastoquinone, and it is transfer that electron to plastocyanin. That's why it is an electron transporter, not an acceptor. Actually it is proton acceptor, that I am telling you just a minute later. Then what happened? Here the process will go on. Now we'll continue after PS2 after some time. So when sunlight fall on the cytochrome, okay, the energy is releasing, and in this in the stroma part, there was proton ion are present. Okay, this is 2H plus ion are present. So when the energy passes to cytochrome, then what happened? The cytochrome, when this electron passes from cytochrome, so electron will going to release energy. Okay, it will going to release energy, and the energy that was released by this electron, here is the pair of electron. Okay, the energy which was released by the electron is utilized by cytochrome to accept this proton, and they will accept this proton from the stroma and pass this, they accept this proton from stroma and pass this proton into the lumen. Okay, one thing happened, that electron was accepted and it passes to the lumen. From the stroma it is accepted and it passes to the lumen, and that's why I am saying that cytochrome is electron transporter and proton acceptor, because it accepts proton and passes it to the lumen, and proton it take proton from the PQ and transfer it to PC. Now one process, now here is the deficiency of electron, so the photolysis of water will occur. So if photolysis of water occur, the three thing is released during the photolysis, we all know that, and that is a pair of electron is released, then a pair of proton is released, then the third thing is a molecule or half O2 is released. Okay, these three products are released during the photolysis of water. This electron will goes into the photosystem to compensate the electron loss. Okay, this O2 will get diffused out from this thylakoid by the process of diffusion, because this oxygen is the byproduct. Now what is left? That is the proton. Okay, so different type these was the process will go on, and the proton will release by the splitting of water, and also due to the transport of electron, the proton will enter from stroma towards the lumen. So what happened? By this process, what happened as a result of these two processes? The concentration of proton will increase inside lumen, and its concentration decrease outside thylakoid, that is in stroma, because the proton is continuously coming inside the lumen. That's why it is become deficient in proton. Now a gradient is developed, that is a concentration gradient is developed, that is proton is more inside as comparison to the proton that is present outside. So now to compensate this deficiency, these protons want to move out from the thylakoid and go into the stroma. But for going out into the stroma, they have only one channel is present. I am rubbing it out because I am making one. So only one channel is present. This is, and that you have studied in previous chapter, that is of F1 and F0 protein. So there are two type of protein are present. One is this embedded or integral protein that is embedded inside the membrane, and it is called F0 protein, and a protein which is present outside the membrane, and it is called F1 protein. These two protein are present, and F1 act as a ATPase enzyme. Now these protein are present in membrane, and the channel to pass out proton is this only, by the help of facilitate diffusion. So what happened? These have high energy, so these proton will come here and they will enter with the energy. Okay, so they will enter with the energy into F1. I am showing it wavy line, that will show you the energy of proton. Now the energy that is present in proton get utilized by this F1 molecule, that is ATPase molecule, and it utilize it to fix one IP, any inorganic phosphate, and one ADP, and ultimately by utilizing this energy it will convert it into ATP, and thus the formation of ATP take place, and now the energy is released out. So the energy level of this proton will goes down and it become normal in condition. So now the normal proton will move out of this and come into the stroma. Okay, it will come into the stroma. This normal proton will come into the stroma, and now like a wave, one by one the proton which is present inside will go outside with the help of this complex, that is F0 F1 complex. Now what is happening? This is the first step. Now next what is going on? This PS1 is again doing the process, that is electron will accepted by the primary acceptor, then they will release energy and they will go into the electron transport chain, and first member that will accept electron was after primary electron acceptor, that is FD, that is ferredoxin, and after ferredoxin it is passes to cytochrome complex, and then from cytochrome complex the release of NADP occur. So what happened here, the NADP that release will bind with this NADP reductase enzyme that is present at the outer membrane toward the stroma, and now the reduction has to be take place. So where the electron that is needed, the proton that is needed for it will go from stroma into this. Suppose this is the, and this will go here, and this with the help of this proton and this enzyme NADP reductase, the NADP get reduced to NADPH2. This is the process that will occur. Actually, the formation of ATP occur in this manner, and this is the original process by which the formation of ATP occur, and how the reduction of NADP occur, and how the process will continue. This is shown here. Now for remembering this, we have to remember that the site of water splitting is lie on inner thylakoid membrane. Second thing, we have to remember that site of NADP reductase enzyme is present on the outer membrane. The third thing that we have to remember that lumen contain oxygen which will diffuses outside. The formation of oxygen will take place inside lumen. And fourth chीज that we have to remember, that is the F0 F1 complex that will act as ATPase enzyme, and that will help in a formation of ATP. So this is the chemiosmotic hypothesis. Due to the concentration gradient, the formation of ATP take place, and this is how the formation of ATP occur during the non-cyclic process. So next we are going to study the dark reaction. Now the light